Okay, I think this is everyone for now. Then again, hello everyone. Have a good evening, morning, afternoon, wherever you are. Today's talk will be by Niklas Bücker from the Friedrich-Alexander University in Erlangen-Nürnberg in Germany. And he'll be talking about paleoenvironmental reconstructions using the abrasion of microfossil fish scales and teeth. So the format of today's seminar, just to remind you, or if you're new here, we'll start with the welcome and announcements. That's what I'm doing right now. We'll then have Niklas' talk, which will be around 30 minutes, which will be followed by a moderated Q&A. And after that, Niklas has agreed to stay for tea time. So if you want to chat with him afterwards or want to ask some more detailed questions, then you are very free and welcome to stay afterwards. So a bit more housekeeping. PaleoPerks values the participation of all folks interested in the paleosciences. Please remember to abide by our code of conduct during today's seminar. Also, please mute yourself during the duration of the talk out of respect to the speaker. You should not be able to unmute yourself, but if you do find yourself unmuted, please mute yourself again. During the Q&A, you can ask questions by chatting to the questions at PaleoPerks host, which is Isaiah today. Or you can also use the raise hand function if you want to ask the question yourself. Also, if you have any technical issues or questions, you can ask them to Isaiah at the questions at PaleoPerks host. We also have closed captions built into Zoom, and you can use the CC button to either show or to hide them. We're also always looking for new speakers. PaleoPerks exists because we have so many cool speakers that are willing to share their work. So if you know a cool early career researcher, or if you are one and you would like to share your work, then please nominate someone or yourself. The link will be dropped in the chat shortly. And then finally, we always do a weekly feedback form for demographic info. This is completely anonymous and completely optional, but we really encourage it because we want to make PaleoPerks as interesting as it can be for you. And for that, we need some feedback. This would also be dropped in the chat. Then enough about us and a bit more about the speaker, Niklas Bücker. Niklas did his bachelor's in geosciences at the Goethe University in Frankfurt am Main in Germany. And he's currently doing his master's in paleobiology at the Friedrich-Alexander University in Erlangen-Nürnberg, also in Germany. Aside of that, he is also an assistant scientist in paleontological curation at the Senckenberg Institute in Frankfurt am Main. And with that, I would like to invite Niklas to share his screen and to start his presentation.
Thank you very much for the invitation again to let me speak here about one of my research topics, which is about the reconstruction of paleoenvironments using the abrasion of microfossil fish scales and teeth. And this talk will be mainly about my bachelor thesis and my upcoming master thesis. So bear with me to not see too many results, but if you have ideas, questions, answers to my own questions that will come up during my talk, I'm very happy to chat about it. A little bit more about me. I'm a graduate student in Erlangen at the FAU. And my research interests are very, very broad. Basically, everything that got bones and sediment in it is interesting to me. But I'm mainly focused on taphonomy, bone bed dynamics, the reconstruction, or the research basically on paleo ecosystems. And in terms of animal groups, I mainly focus on fish, Cenozoic mammals, and dinosaurs at the moment. You can see some of my research topics basically and myself during an excavation in Frankfurt actually. I want to give you some background to my bachelor thesis, or to my thesis, because I quickly realized that not too many even paleontologists are familiar with my main focus, which are bone beds. The bone bed itself is very hard to classify or to even determine what it basically is. But the first mentioning was in 1834 by Charles Lyell, who called or described a deposit at the Faroe Islands near Ireland as a deposit composed of fragments of teeth and bones in extraordinary manner. Very broad. It was a recent deposit, so no fossil one, but basically a lot of bones, a lot of teeth. So very, very, very broad. A little bit later, or over the years, more definitions and approaches came into play. And one of the more important ones, I would say, was from Antia, 1979, which tried to describe or to describe bone beds in a more chemical way, which is based on the phosphate contents, which from 4.5% on is you could call it a bone bed, or you could consider it a bone bed. That will be more important later for my own samples. And Behrensmeyer in 1999 did go back a bit to the pure bone content itself. So basically a sediment or a strata layer that contains a lot. Here 5% by volume, so not that much actually. You could call a bone bed. But the best fit that at least for my thesis, came from Rife in 1971, which is a vertebrate sand. It's technically not really a bone bed. It's a new term, but it describes what I have analyzed pretty good, which is a sediment that contains disarticulated and abraded vertebrate remains that show marks of heavy relocation only. That means there's basically no really fresh material, nothing that got deposited very recently in terms of the diagenesis, but rather like a lot of stuff that got transported, reworked, and so on. And with that, I want to go back to my research topic itself, the Ratian bone beds of Europe, specifically Germany, but in all of Europe, they can be found in France, in Poland, Luxembourg, Netherlands, Serbia, and also in Germany. Some of these bone beds are not accessible anymore. For example, in the Netherlands, there was a construction site that got sampled on, and then a house got built on it, so not accessible anymore. But you can find them pretty much everywhere in Europe, and they are actually known for 180 years already, and they are still researched on because nobody came up with a very exciting answer yet about their deposition. In England, or the English bone beds are mostly interpreted as storm event deposits, sometimes multiple events, but not in every case. But in the papers itself, the preservation of the fossils is not really discussed. It's more about the content and the biodiversity, and the amounts of fossils, and not really the preservation. It's barely mentioned, like, oh yeah, there's some abraded teeth and fossils, and some very nice ones, and obviously they show the nice ones in the papers. And with that, we come to my own samples, which come from the southwestern part of Germany, from the state of Baden-Württemberg. And they come around from the northern part of Baden-Württemberg, and the area is known locally, at least, as the Swabian Cloacae, which is a very funny name in my opinion, which is actually just based on the pure fact that in these bone beds are a lot of coprolites, which comes back to the or calls back to the determination of a bone bed based on the chemical contents, because there's a lot of phosphate in these bone beds that is coming from the coprolites. But there's a lot of other stuff in there. Obviously, we have hypodontid sharks, we have Gyrolepis, which is a genoid bearing genoid scales bearing fish. You can see down to the right. Saurictis or Sevenictis, as it's sometimes called nowadays, which is in the middle. Some mammals, but also dinosaurs, ichthyosaurs. Really, these bone beds flourish in biodiversity, depending on the local situation, obviously. And the first interpretation that was ever published by a geologist or geoscientist was by Quenstedt, and he interpreted all the area around my samples basically as a tidal flat. Some of you who either are from Germany or know the northern parts of Germany, they know parts which are called Watt in Germany, which is a mud flat basically. Here it would be a sand flat, because we have a lot of sandstones in here, not really, like more or less no clay minerals or silts, but the general environment would look like a tidal flat or like a mud flat. And that is very interesting, because in the literature, a lot of different ideas came up to interpret these environments. And what I was supposed to do was making a classification, or trying to do a classification of bone beds based on the preservation of the fossil content. For that, I handpicked 200 specimens for each morphogroup. These morphogroups are based on the fish that I showed on the last slide, and I will show them in a minute. And I had 2 samples to compare and to look at them and try to say, okay, is there any correlation? Can you actually even classify anything in there? And these are more or less my results. And you can see here on the left-hand side, this is one sample. On the right-hand side, there's the other sample. And I constructed 4 different morphogroups. On the left-hand side, can you actually see my mouse? Yes, right? Yeah.
Yes, we can see a mouse.
Yeah, perfect. This row in here is the hypodontid sharks, Rhomphiodon, Hypodos, you name it. This is Lysodus, it's a shell-crushing shark. This is Saurictis, and these are genoid scales that come from a fish called Gyrolepis. And the order is basically from the best preservation on the top, going down or getting worse to class 4, which is the worst preservation. And this goes obviously for both samples. And I came up with these, and I classified 200 specimens of each morphogroup and each sample. And then I did a statistical analysis, very, very brief one, nothing related with R, but just pure eyes, basically. And this is these are the end results, basically. And you can see in here the darkest colors, these are the worst preservation. The lighter colors, these are the better preservation. And you can see some trends in here. For example, in the Lysodus and Hypodos sharks, the teeth, the preservation is actually very bad. You only have remains of the tip of the teeth, or just the root, basically. But for the Saurictis, you cannot really see a pattern, I would argue. But for the genoid scales, these are very, very strange, because they actually show the absolute contradict pattern of the teeth, for example. The scales are more or less all perfectly preserved. And this is quite strange because teeth and scales consist of the same material. You can see in here, this is a very nice specimen to explain this. You can see on the scale itself, the light grayish part, this is enamel. So basically the stuff that makes our teeth and also is on the tip of the fish teeth. And below it, there you can see it in the class 3 in here. There is a bony layer. So this is actually bone as in our bones, and on top there's this enamel. So they consist of the teeth and scales consist of the same material, and but we really don't know, or we didn't know why the scales would be so much better preserved than the teeth. So I came up with some explanations, or I tried at least, and searched through the literature. Sadly, there's actually not that much literature in or about ganoid scales or the preservation of especially these type of fossils. Of teeth, there's actually a lot, but more about microwear and not really abrasion post-mortem. But the first solution that we came up with was a very, very simple one, which could be the center of gravity, which would mean that especially the top enamel layer was just heavier than the bony layer, and if you imagine a very flat environment, a flat marine environment, or a shallow environment, that the scales just get turn on the enamel side and then get moved around, and they might get a little bit abraded, but the enamel itself is so hard that the scale is not vanishing, basically, or breaking up. But the problem is, when we looked into the source rock itself, there was no clear evidence for that. So there was no direction or orientation of these scales, which would explain this hypothesis. So I searched further. I thought I had it with the biomineralization, because there's a very slight difference in the structure of the enamel in ganoid scales and also in teeth and fossil teeth. But this explanation is not that good, because it's not really evidence-based. It's more about it could be not. So that also got thrown away. But I might found a solution for that, and this actually comes from modern sharks as well, because when shark skin is drying, and maybe some people in the audience are interested in his history, actually shark skin was used as sandpaper in former times, because when it dries, it gets very, very hard, very, very sturdy. And the same actually happens with ganoid scales. Ganoid scales, when they are fresh, when they are wet, and actually from the living animal, for example, then they are a bit bendy and very soft. But when they get dry, so if you dry them for some hours on, I don't know, maybe 30, 40 degrees or so, they get very, very hard, but brittle, which is the only concern about my solution. Because when you can see in here on the left-hand side, this is from a work from Chen et al. From 2012. They actually measured with a nano indentation, like little borings, basically, how hard the ganoin of this ganoid layer and the lower bony layer is. and you can see a clear difference in here. So on you can see on the left-hand side, this is the genuine, which is very, very sturdy, very, very hard. But then the drilling got into the bony layer, and it is very, very, very soft to see, or it's not hard at least. The problem is nobody did that with fossil genoid scales so far. So last year in October, I reached out to a department of our university, the material sciences, and I asked them, could we do this with my fossil scales? And they were like, oh yeah, we could try that. They're very, very small, very hard to work with, but we can try it. And as you can see on the right side here, there's not really a clear pattern, or not even the same pattern as in the recent scales. So that might be a solution, but we have to investigate this further. And I will do this with my master thesis, which I will explain in a minute. But with that, obviously a bachelor thesis is still a bachelor thesis. So I took this interpretation and I came up with an interpretation of the taphonomy, basically, of the history of the deposition of my samples. And at the beginning, as you can see here, we basically have an accumulation and natural deaths of fish, and the scales and teeth get a little bit transported. They get abraded a little bit, but not too much. And with the tidal flats, which comes now into play, with the wave movement and even sometimes storms or little storms, they get washed onto the beach. And get dried by the sun, wind, temperature, whatever. And with the next tide, the objects get back into the water again, into the water column, and get transported. And the teeth, even though they also get a little bit sturdier sometimes when they dry, they probably don't get as resistant against abrasion as the scales. So the teeth get rolled around based on their own hydrodynamical behavior, but the scales don't really change that much. At least that's what we think. Then they get deposited in a sediment, and on top of the sediment, there's a new accumulation again of objects. And as in the interpretations of the English bombards, for example, storms rework the early deposited layers, and then the whole process starts again until the very end of the Radian, so the latest Triassic onto the Triassic-Jurassic boundary, because you have, especially in the Radian itself, you have a lot of fluctuations of the seawater level. And with a last big and slowly going on transgression into the Jurassic, then you have the final deposition of the Radian bombard as we know it from Germany and also different places, which is at least or would be a good explanation for also the geology, which is highly complicated in Germany. And with that, we will take a little look into the future, because this is like the basis I work with now. But obviously there's a lot of research still needed, because, for example, we could actually find evidence for drying of these genoid scales in the bony layer, because you can see on here on the right side, these are pictures of the bony layers of genoid scales from recent alligator gars, and you can see these little dots in here, these little black dots, and these are actually channels that transport nutrients into the surface of the scale to build up the enamel layer or the genuine layer. And interestingly, when the scale is still wet, if they are cracking up or if they are breaking, you can see the break is more or less irregular. But interestingly, if you dry the scale and then if you then break it, you can see the little dots in here, and they actually follow these channels or these tubes of the bony layer. I didn't have the opportunity until or for now to actually investigate this in the fossil ones, but it would be very interesting to analyze. And I really hope that I'm able to do this for my master thesis as well, because that could at least be or support the hypothesis of drying of these scales. Obviously, I want to do some field work and observations of new sites, so either and also collections, obviously, because there are a lot of specimens laying around in museums and collections, especially in the UK. But in Germany, for example, there are still a lot of possible locations where you can find these radium bone beds, and I wouldn't want to do or to apply my methods on new locations, obviously. But for my master thesis, I want to do some experiments actually, because I want to at least find more evidence, or even or obviously also disprove my own theory for my bachelor thesis with using tumbling machines. I want to simulate basically the environment, different environments where these ganoid scales or these fossils may have lived or were present. So I don't know, fresh water, for example, because we also have a lot of terrestrial influence in the radium bone beds, a shallow marine environment with different grain sizes and different salinity levels and stuff. And then I want to want to simulate the transport basically for maybe some years. Like the simulation should go for some weeks, but to simulate multiple years of transport and then look at the specimens again, if they show the same patterns as in the samples I analyzed. And I have a little trigger warning because just to be safe, on the next slide, there will be shown a carcass of a fish. I will say if this slide is gone, just so if you don't want to see that, you have to look away for some minutes. And obviously, I want to work with recent scales. So like 2 weeks ago or 1.5 weeks ago, I was able to go to Stuttgart into a zoo in Germany because recently one of their alligator gars died, sadly. But they put it in the freezer because they want to take the head or the skull for education reasons. And I was allowed to take some samples from the scales. They are now in my freezer, and I will use these for my experiments. I also want to use, obviously, my radiant samples and put these in again and look if they also behave in a certain way. So you can see actually the difference between recent material and fossilized material. And also because the scales are so well preserved, I want to take samples from the messel pit from the Eocene. Luckily enough, my supervisor works there, so I will get access to the scales. And I want to throw different, a small amount of scales into every tumbling machine and look how these behave. And hopefully test my interpretation in a positive way, but obviously you never know how fossils behave, especially not in experiments. And if depending on the outcome of my master thesis or the experiments, I want to continue on, as I already said, other regions. For example, in Germany, here on the right-hand side, you can see another radian location near Bohnenburg, which is in the in northwestern Germany. And I was allowed to be part of an university excavation last year, and we found a lot of ganoid scales and teeth, and they were actually very nicely preserved, which would support my idea, basically, because these are all clays. Not like they're not sandstones like in the southwestern part of Germany, but clays and silts. And they are a bit younger than the samples I collected. And I also, because I'm working in the Senckenberg Museum, and we got a small collection of ganoid scales there as well from the late Maastrichtian, so the latest Cretaceous, from Wyoming. And I want to investigate the scales there as well, because we have a bone bed there with different layers, one sandstone layer, an intermediate layer, which is more or less both, and also a mudstone layer, which is interesting because I already did take a little look into the specimens, and the preservation matches my interpretation pretty good, because in the mud flat or in the mudstone, the preservation is way better than in the sandstone, actually. And there, by the way, the carcass is gone. I forgot about this. So you can get back again. And actually, with that, I'm more or less already finished. I want to thank my supervisor, Philippe Havlik from the Messerpit, Dr. Michael Wurmshuber from the Material Sciences here at the FAU, and Miriam Wüst, a colleague of mine from Stuttgart, from the zoo, who I got in touch with for my recent scale samples. And if you have any questions, if you have any ideas or comments or criticism for my work, please tell me, because I during my bachelor thesis, or during the I got a little bit insane because I just couldn't come up with proper ideas, and because there's so few research on these specific topic, and I hope to come up with a solution. But obviously, a shared swarm mind is way better than just one brain or two brains. So yeah, thank you very much for listening. And if anything is unclear, obviously as well, just question me. Yeah. And I think I will share.
No, please
yeah.
leave the slides up. But
Okay.
thank you very much, Niklas.
Yeah.
That was a great talk. I think it's very cool to see this experimental paleo work. Just a reminder for the audience that you can send questions to us anonymously, or you can also put them directly in the chat if you don't want to stay anonymous. We have a few anonymous questions, so I will just drop them in the chats.
Mm-hmm.
So the first one asks, what are the main drivers influencing the preservation quality of the teeth and scales? You discussed a lot about the abrasion, but maybe like the other factors that play a role.
The abrasion is probably the most important factor, because there are different types of shallow marine environments, basically, to very cut it down to the most important part. There is a high transport or low transport environment, and low water energy and high water energy. So you can actually have, like, for example, if you have a storm, usually the transport distance is not as long as in very calm environments. And that we actually saw in my results. So in one of my samples, probably the transport distance was a bit longer. So the transport before the final deposition did go on a little bit further than in the other sample. In the other sample, the energy of the environment was a bit higher, but the transport was less. So that might indicate storms, but we are not sure. But obviously, for the pure preservation, there's also, you have to take into account the basically the wearing of the teeth, so or also the scales. So if they are used in life and the fish is dying, obviously there's a little bit of rounding, but you can, if you look very closely, you can determine either a wear of these of the teeth, the tooth, or if it's really abrasion, because you can see little ridges on the tooth, and if they are present, then it's mostly usage, and if not, then it's abrasion, and that is the factor that we found the most in the teeth. I hope that answers the question.
Thank you. I think so too. Otherwise, the person who asked, please get back to us. Maybe related to what you said in the end, the next question asks, why are the scales often better preserved than the teeth if they are made from the same material?
Yeah, that is like the question I'm asking myself since 2 years already. They are more or less the same material, but we think it's based on the hydrodynamical behavior and also on the extended hardness that comes with the drying of the scale. So if the scale gets flushed or transported onto the beach, it gets dry, it gets very, very sturdy against abrasion, and then it goes back into the water and at some point gets deposited and fossilized and through diagenetic processes. But the process itself is very hard to investigate because there's no research on it. Nobody looked at the preservation of these fossils and how, like in what way they preserve. So we have to do some very basic research. And then hopefully after my master's thesis, I can answer this question more clearly.
That's a good answer. Sometimes you need a bit more time and data. The next question asks, let me put it in the chat. Cool talk. Could some of the teeth have been transported much further than others, depending on the habitats of the species? I don't know how well constrained these habitats are.
Yes, definitely. Because you have like brackish environments, you have shallow marine environments, you also have open sea environments in the Radian, and the teeth could be transported further. But the interesting part is that and for that the roots and the breaking behavior of the teeth is also very important, because you can actually distinguish between the pre-fossilized breaking, so just before the tooth was fossilized, it breaks. You can see that in the fossil in the end, and you can distinguish that from a breaking post-fossilization, and we've found both. So we think that is evidence for a reworking of the early deposits, so that the already fossilized material got reworked again, breaks, gets transported and abraded even further than before. If you speak about the habitat as, for example, freshwater, and that got flushed into the sea, that is a bit harder to determine, I would say, because the good and the bad thing about these fish is that they live in freshwater and also in marine waters. At least they did in the past. So we find terrestrial material, so there might be some influence from the terrestrial as well. But so far, there's no possibility to determine that in a bone bed. but it's a good question. And I hope to answer that as well sooner or later.
Thank you. Then we have a question on all this material that is maybe in museums. So someone asked, thank you for the interesting talk. What about the sites that are no longer accessible, like those in the Netherlands? Was there enough material collected to do similar statistical analysis?
Yes, definitely. More or less every like for the Netherlands example, there are multiple thousands of objects found in the bone beds because they are so small and you don't need that much source rock basically that you have to prepare. So it is possible, I think. Obviously, I want to increase the sample sizes further. 200 is okay, but obviously not too much. But for the UK, for example, there are like very, very large collections. I'm not so sure about America or like different times or different periods, but I know for sure that the collections that are currently available or described in papers, they are large enough for that. And we also want to work or train an AI to automatically classify the material so you don't need that much human power basically because it took several days or weeks obviously to classify everything, and when you increase the sample sizes that gets even harder, but it is possible. I just obviously need access to these collections.
All right, but so far there is no one or very few people working on these collections for this purpose.
Yes, for this specific purpose, there's basically no one. I found one master thesis who worked with the preservation of ganoid scales to use them as a paleothermometer. That didn't work out, sadly. But until now, it's more about the biodiversity of these bone beds and how this changed over time, and not about the preservation itself.
Okay, interesting. So there's still a lot to do.
Yeah, as always.
With that, so far we are at the last question, which I will drop in the chat now. So I'll remind the audience, if you have any further questions, please send them in. And this question asks, could we use the information on the degree of drying of the teeth and scales to say something about sedimentation rates or subaerial exposure time if we have enough data?
I would say yes. But the problem with bone beds is mostly that they are very unique for their locality. So to come up with a overall pattern for every bone bed is very hard to do. The sedimentation rate, I would say, it could be a bit harder because of that, but the subaerial exposure time could definitely be reconstructed, I think. That's why I want to do, or that's why I want to dry the recent scales as well to different degrees and for different times, to at least say, okay, they might got exposed for like days or weeks or just hours, who knows. But I think that is definitely possible, yes. Yeah.
Okay, thank you. Interesting. If there are no further questions, I have one final question myself. On the more chemical side of diagenesis of these things, let's say, like I think that most of the deposits are in more sandy deposits, but I think near the end you mentioned something about mud. Could there be any influence of microbial degradation or anoxia versus oxic sediments that affected preservation of these skills?
I would say yes, at least in the coprolites. It could actually be that the like the stomach acid that is still in the coprolites abraded the teeth in the scales, and then the coprolite did fall apart because obviously there's a lot of fish poo. But I would, if there is microbial impact, then we don't have any evidence for it. At least not in these, like not in the sandstone bone beds, because there's no clay, no heavy minerals, no nothing basically, just quartz and fossils. So, but if it would be actually interesting, maybe if we come up with a technique that maybe with thin sections, that could be a thing, actually, because the sandstone or the samples I worked with, the source rock is very hard and very it's not easy to prepare for the investigation I did. So the microbial traces might get lost because of that. But in the source rock, it could be an interesting idea. Yeah.
Okay, interesting. Then with that, if there's no further questions, I would like to end the Q&A. And thank you again very much, Niklas, for a very interesting talk and very good discussion in the end, I think.
Thanks for having me.
Yeah, no problem. Thanks for joining us. Before we go to tea time, I will share some info about next week's talk.
Yeah, I will stop. Oh, yeah.
So yeah, thanks everyone for joining us. I hope you also had a great time. Again, a reminder to please fill out the weekly feedback form. It will be linked to the chat again. We would really appreciate it. We hope that some of you will stay for tea time, but if you don't, then we hope that we see you next week, October 14th, same time, where we have a talk by David Kirk from the University of Sun Yat-sen in China, and he will talk about acritarchs and their role in biostratigraphy. So for those of you that don't stay, I will hopefully see you next week. For those of you that want to stay for tea time, that will be up next. So tea time is just an informal chat with today's speaker where everyone can turn on their microphones and their cameras and just have a chat. Or also if you have maybe a bit more complex questions that you couldn't ask during the Q&A, then you can ask them now. So before we do that, we have a quick 2-minute break. So also the speaker can have a pause. And after that, I will hopefully see you again. So see you in 2 minutes or next week.